This is a great venue, very suitable for long reads. So we're going to start here. So I'm going to walk you guys through the challenges that we went through developing an assay for adventitious agent detection using NGS. And so here we decided to go with long read because we think that detecting an adventitious agent with only one read of like 500 base pair or more is going to be easier than just 150. So that's why we went for this, and so we started developing this. And there were some difficulties with detection of high background matrix sample, and so we're going to go through all these aspects of this development. So the case I'm going to present here, so the method that we are using, I'm going to be clear on this one. We use the 48 hours here for sequencing because we noticed that above 48 hours, the data doesn't bring really added value there. And so we limit that to 48 hours. We choose to go with the ligation kit in order to have longer reads. We tried the rapid kit. It was very rapid, but the read was short. So we didn't go with that. And in this case that I'm going to present you, we tried to skip the extraction, the nucleic acid extraction, and went straight with like heat extraction, which is like you heat up your sample, burst open the virus, and then access the nucleic acid. And then we added to this because when you go with heat extraction, the volume is not that high, so we decided to do a little PCR to amplify the nucleic acid before doing the sequencing. And so here's the bioinformatics analysis. This is something that came in kind of later because we had to take time to kind of develop this. So pretty much the workflow is to really generate we generate the FASTQ file, and then in that bioinformatics pipeline, you will have a quality control and then some read filtering, and then you have the read alignment with the genome database, and we also do a read alignment with a protein database. So that those two, when they kind of align, we know we have a real hit. And we have some criteria that we define there from, to accept a positive read there, which is like the genome coverage that we are requiring. Minimum has to be 10%. And then at least one ORF in that sequence that we're going to analyze. And so that's the flow. So for this case, we use the Advitig sample. If you guys are not familiar with Advitig, it's a consortium run by the FDA where it's free. There's a lot of biotech, and we kind of running study and performing those studies at each lab and then comparing results and try to come up with some consensus kind of testing there. And so in this spike study, there's viruses that we want to use to do this study, and those viruses are now called the WHO standard, so they're available to anybody to kind of purchase and do some study there. So what we have done here, we kind of did two prep. One prep that you call the sample A here is going to be the one where we're not going to spike anything to those sample. And the sample B is going to be the one where we're going to spike an internal control, which is MS2 phage, that will come and then add additional nucleic acid to this. And the reason why we wanted to add the phage is to see if there's any inhibition in that process, especially when we do amplification. And so the experiment workflow, we do sample prep, we do heat extraction, universal PCR, we do the sequencing, the library prep, and then we do the sequencing on a GridION, and we do the analysis. So this is summary results here. So as you can see, this is a summary for sample A where we didn't spike anything. We have been able to detect all the virus spike, but there were some additional viruses there that we found. Bosa virus, the squirrel monkey, and the human ERV K113. The Bosa virus and the squirrel monkey are believed to be contaminant from those virus spike that we purchased. And this has been confirmed with colleagues at the Adventitious. And the human ERV is, we suspect this one to come from reagents that we use there. And so as you can see here, when we go with the spike level that's lower at 1E4, the first one is 1E6 for all the viruses in a background of 1E9 of adeno. And here with 1E4, we start losing some viruses like PCV and FLV. And then we noticed that there were some reads that we detected, but we couldn't confirm those reads with the ORF alignment. And so we said that maybe the LOD is around that level there. If we go with the detail, you can see here number of reads that we are getting. And so this will be for the sample that spike at 1E6. And yeah, it's pretty good read. And then we have HPV here. You see 68 read. We have been able to show that there's one ORF for that HPV. But again, it's a contaminant from the virus spike that we had there. The A2, which is the 1E4, we start losing this, and so forth. And one sample here, which is the actual adeno that we use to spike for the background. So when we did that, we can see that adeno here. And one sample that really surprised us is the negative control. So we use here negative control, which is just water. And you can see here we're getting some reads from the adeno and also from read from, like I said, the human, also ERV and HPV there, which is probably coming from the reagent. And we suspect that we have cross-contamination with the adeno at 1E9. And then we did some testing in the lab. We noticed that with AAV, when we have AAV exceeding 8 log per ml, we're getting the same contamination with the negative control. With the sample B here, with this time we add MS2, right? MS2 as a phage to control really the experiment and make sure that the RT PCR is working, the PCR is working, also the sequencing is working. And here we have been able to detect our MS2, and same thing here, we lost FLV again at 1E4, but this time we have been able to detect PCV. So PCV must be at that borderline there at 1E4. And also confirm the detection of the other contaminant like Bosa virus and squirrel monkey and human ERV. If you go into detail, you can see here at the sample that spiked with 1E6, MS2, our internal control phage, is showing up at 246 reads only. And if you look at the sample B, where we spike less virus, MS2 is showing up at 1,000. So this is showing us that you have some inhibition somehow, but it's not complete to inhibit everything. And you can see here, when we go with just one virus, the adeno, MS2 is showing up at really its full level here. And so this is also now the water sample. When you see the water sample, we're still having this adeno contamination into the water. So this is really a challenge that we are going to have with testing samples that have high background. If it's in the vaccine will be high background. Vaccine might show up into the negative control. Or if it is a viral vector, the viral vector might show up into the sample. So I have 9 minutes here to go through the challenges. So the point one to consider here is going to be validation. Validation of the assay, what virus model are going to be used for adventitious agent. Here, the problematic is not to detect only one virus. We're trying to detect all of them. So what virus are you going to use to show that the assay is capable of detecting all of them? This is something that we getting discussion with the Adventitious group to have those WHO viruses pool that are kind of encompassing any kind of genomic structure of the virus, like single-strand DNA, double-strand DNA, circular DNA, double-strand RNA, single-strand RNA, negative, positive. We have all these kind of virus representative in that pool. And that's the pool that we were using. And then you have your instrument IOQ. So we had the research line, so now we're switching to the Q line because we kind of encountered some challenge for the IOQ, for especially data integrity and audit trail. And those have been resolved into the Q line, so now we're getting our Q line up for shipping. And the pipeline software validation. So this is something also, it's going to be a big challenge there with cloud computer validation. And also, how do you kind of maintain that software or how do you deal with the change control when you have to change, for example, the database? Database gets evolving. Database is not complete. Maybe you will have a new version of COVID that will be added to the database. You have to update your database. So we need to have kind of a change control system for this. This is something that we also putting together. And Part 11 compliance, the speed and cost of analysis. This is something that will come to you at some point. You can do very nice R&D. You can put your test together. If your test is very expensive, it's going to add more cost to your product at the end, to your medicine. So this cost is something that will be important at some point. The sample preparation is something that we have to work on depending on the sample type that we're having. Here, we just went with like a proof of concept with virus put together into a buffer. So that's not really representative of other sample that we might be testing. So there will be a sample prep that there is a sample preparation that we develop in-house that will be applicable to kind of cell culture fluid and other type of sample. This is meant to reduce the background so that testing can go with higher sensitivity. Nucleic extraction or not, in this case, we didn't do nucleic extraction, but we are planning on doing nucleic extraction so that we can increase the volume and concentrate the DNA so that we can hopefully increase the sensitivity here. Pre-amplification or not, we're generating data with pre-amplification and no pre-amplification, no PCR. PCR tend to give us more reads, a little bit more sensitivity so far. But if we can skip that step to reduce the contamination, is something that we might consider as well. A comment on that one, if you're using the Prometheus, Prometheus has more sensitivity than the grid ion, so maybe on that one you don't need pre-amplification. Number 3, internal control and negative control. This is a QC assay. You're going to need a negative control and internal control to really have a full control of your assay. So now we see some colleagues using like 2 phages, one RNA phage and one DNA phage. Yes, but here we judge that using only MS2 as RNA phage allow us to control the assay from end to end, because the RNA phage, after your RT, you're going to do PCR. So if you have inhibition, your PCR would be visible right there. So we don't think that you need to have two phages into your assay. The sample background is something that's going to be very challenging here, but it also, even if you reduce it to some minimum, you're always going to have the sample background. So we were kind of analyzing here if that sample background can also be used as a DNA control. The negative control will be contaminated with high background. Be ready for that. And so for this one, for this type of study, what we're doing, we kind of lay out the process, and then we did kind of a risk assessment of each step and see what is the highest risk of cross-contamination each step and provide a mitigation for those. So bottom line is to work under a clean room and where you have control of the environment so that you don't contaminate amplicon or genomic sequence. The criteria for positive and negative results. So here we have a system suitability with the Q-score and the negative control. The Q-score is not something that we really give a huge weight about. We just say, if you have your Q-score above 10, it's something that will be sufficient to determine if you have an adventitious agent or not. So here, negative sample is defined as reads where the coverage is below 10%. Really, 10% is nothing. It's like if you have a genomic, small genome virus like parvo, you're going to have like 4 kb as a size for the genome. So 10% of that, we're talking about 400 base pair. It's really no big deal. And then we're also requiring to have, for being negative, to have 0 ORF. So we have a sequence, that sequence, no ORF. We know it's like a piece of genome somewhere. And sample suitability for the unmapped read, we want to have like less than 1% unmapped read of the total. For the positive, we want more than 10% coverage and at least 1 ORF. And above this, we want to do a confirmatory test if we do have a positive. And the confirmatory test will be extract those sequence, design PCR, and look for the virus into that pool. When we see sequence like bacteria, fungi, and we do see them, we tend to put them in the pool because this is not in scope for that testing. That testing is meant for detecting virus. Here, we said that we need to account for all reads because the risk here is that if you have like 10% unmapped read, what if that 10% unmapped read is a new virus that was not in the database? So that's something that I think needs to be addressed during validation and ensure that you always have a minimum unmapped read into your pool. The negative control exhibiting sequence, that's a big challenge here. Like I said, we going through some experiment with a lab setting, equipment setting, reagent control to reduce that and make sure that we have a negative control that's truly negative at the end. Bioinformatics is going to be your big challenge. How do you do the analysis? Analysis turnaround, we want like 24 hours for the bioinformatics, and alignment accuracy, if using Kraken, not very accurate, use Minimap 2, maybe a little bit more accurate, and computing resource like memory is being an issue, especially when you're doing genome assembly, it's going to consume a lot of memory. And optimal database, partner and compliance, full automation. Thank you.